I'm Emile. I'm the president and CEO of BlueRock Therapeutics, and I'm really pleased to tell you our story. I believe I hit the green button here. Must be this one. BlueRock is an engineered cell therapy company, and our mission is to develop authentic cellular medicines for devastating diseases. Specifically by that, we mean diseases that have been labeled intractable by the healthcare system. Diseases like heart failure, where there's 34 million people worldwide today, and we have no disease-modifying treatment. The disease course is a slow decline to complete heart failure, 34 million people. There's 8 million Parkinson's patients for which we have no disease-modifying treatment. The standard of care was developed in the 1950s. So we have these profound needs in the healthcare system. These have been problems that have been intractable to existing approaches. We believe we have a new approach to that. We call that Cell plus Gene. We developed a novel platform for this, and we're focused on 3 therapeutic areas: neurology, cardiology, and immunology. Specifically, we use an induced pluripotent stem cell, to manufacture the authentic terminal cell that you find in your body. We intend to replace that cell in the degenerative condition, restore the native biologic that was lost, and reverse the deficit, reverse the disease to provide a functional cure. But because these cells that we manufacture and that we intend to engraft are manufactured from a pluripotent stem cell bank, you can engineer that bank and thus the terminal cell for enhanced function. That gives us the ability to add payload to alter the safety and efficacy profile of the cell, the terminal cell, and we believe that gives us the opportunity to treat rare and chronic disorders in a way that really has not been possible or practical before this approach. So broadly speaking, and please excuse the cartoon here, but this is the way I think about what cell plus gene means. So you have, say, 200 therapeutic target cell types in your body. There are, Wikipedia would say 308, I don't know, pick your favorite number. There are hundreds of cell types. They're all subject to disease. So if you say there's 200 therapeutic cells of interest, there's 20,000 genes in the genome for which they would be of interesting therapeutic targets. That represents 4 million therapeutic opportunities, for which the only way to access it is to have both industrial control and commercial rights to both of those axes. And there have been pioneers in this industry. We all stand here today because of things like the advancements in CAR-T, where we have a single cell type and a single engineered gene at one intersection point. What I'm arguing is there's this immense matrix that requires both axes to access it. So for us, that means accessing the 200 cell types using an induced pluripotent stem cell platform. We have a proprietary platform I'll tell you about to access the developmental code. And the genome code we access through CRISPR-Cas9 editing in our partnership with Editas Medicine. This gives us the ability to, in theory, manufacture any cell type in the body and edit in our therapeutic areas any gene we want. And we believe we are the first company to be so situated with those broad rights and those capabilities. So I think you may have seen a press release about a month ago or a month and a half ago announcing our acquisition by Bayer. We're very excited by that. The purchase price of implied of $1 billion in upfronts and milestones. The reason that I'm excited about it is that it provides us access to the world-class expertise of Bayer, to all of their capabilities, a shared vision on cell plus gene and where we want to go. It's also their vision, but it's a completely independent operating model, so we believe gives us the best of both worlds, and we're very excited to deliver on our commitments. So the platform starts with a material from a donor. We believe we start with the best material in the industry. I'll tell you about that in a second. We've put a lot of thought into what it means to have clinically relevant starting material for an IPS derivation process. You make an IPS cell, you can then engineer it for HLA and other targets so that it is in theory allogeneic and universal. And then you can expand it indefinitely. It becomes an exhaustible, a master cell bank, which I'm sure you're familiar with. This is the basis for the IPS platform. We have put an enormous amount of effort into what it means to have the appropriate starting material. To my knowledge, this is the highest standard that's been achieved yet for commercial grade, for any commercially available IPS line. Complies with donor eligibility requirements globally, testing genotype and extensive pathogen mutations, informed consent, right to recontact, commercial rights globally. We've thought a lot about this. Similarly, we have a proprietary reprogramming process that the IP is exclusive to BlueRock. It's non-integrating and RNA-based. It gives us and our partners complete regulatory insulation, meaning that you don't have to deal with what others missteps. And it's fully implemented in our GMP laboratories. And finally, we have multiple strategies for HLA editing. I think you may have seen our announcement with Universal Cells a year or two ago. Similarly with Editas Medicine, a strategy, and we have internal strategies. So we intend to build a truly allogeneic IPS master cell bank, a universal master cell bank that could in theory be used for every patient in this room. Second, you can engineer those IPS cells for payload. I've already mentioned this, but for safety and efficacy profile, tunable expression, safety switches, enhanced function, and to deliver payload like this, functional proteins, antibodies. Some of the cells that we're developing would be remarkably good at carrying in therapeutics payload into the body and stably delivering it. And core to BlueRock and bedrock to BlueRock is our founders' expertise in turning an IPS cell into a terminal cell. And I'll introduce you to the founders in a moment, but our 3 therapeutic areas, neurology, cardiology, and immunology, where we have this world-class, we think best-in-class expertise to turn, for example, an IPS cell into an authentic midbrain dopaminergic neuron, or a ventricular cardiomyocyte, or a true macrophage or T regulatory cell. Our founders are people like Lauren Studer and Vivian Tabar, founders and founding investigators that have been with us from the beginning. Gordon Keller and Michael Laflamme, again from the beginning, as founders and founding investigators and critical to our knowledge base and expertise. And then new additions like Bruce Blazar and Courtney Crane, who bring immunology expertise in Tregs and macrophages. Those people together have taught us how to make that authentic cell. We've applied this idea of this cell plus gene platform broadly to these 3 therapeutic areas. In neurology, we are manufacturing midbrain dopaminergic neurons derived from the authentic floor plate, and those we will use in a Parkinson's trial I'll tell you about. We have microglia, which we're deriving for neurodegenerative disorders, oligodendrocytes for hypo and demyelinating conditions, and finally, enteric neurons for a disease called Hirschsprung's, which is a gut motility disorder. It's a crushing rare disorder in children. In cardiology, we make ventricular cardiomyocytes and nodal cells for pacemaker type applications, and heart failure for the ventricular cells. And in immunology, we have programs in macrophages and T regulatory cells. So our founders have taught us how to make that authentic cell for the first time in a dish. And what we try to do is to become the best at turning that into industrial expertise and to manufacture that at a scale and at a quality that's relevant for therapeutics. An example I'd give you is Lauren Studer, who in 2010 and 2011 published the definitive method for how to make a midbrain dopaminergic neuron. It's widely cited as the Studer method, and I think it's fair to say is the standard and the right way to make that authentic cell. You can make, using this method, a highly pure population of floor plate-derived midbrain neurons, very low or no contaminating cells. These cells will spontaneously burst in a petri dish. You can see that plot there of the spontaneous bursting profile, which is what's happening in the midbrain of every person in this room, and what allows you to control motor function. The connection between thought and action is this tiny motor center in your brain that controls, that's bursting like that. And these cells, for the first time, showed robust survival in animal models, reversal of the motor deficit in animal models, and we're engaged with the FDA to start a clinical trial using those cells. So I think we've taken this academic expertise and the science of the founders. That still leads, and we know that we can make the authentic cell. The question is, can you make an authentic medicine from that? And there's a big difference between that. To make it at scale, to make it reproducibly, to be able to test it for safety and potency, some significant challenges to all of us. So what I thought I would do is just hit on one approach we've taken to address the concept of scale, potency, and safety, and how you might be able to come up with more rapid protocols and tests, approaches to address these basic needs that I think everybody in this room who's using a pluripotent cell at least would have, or even cells in general would have a similar challenge. So the first is, it's one thing to make 1 million highly pure midbrain dopaminergic neurons. It's another to make 10 billion at the same, in that same scale and that same control. So we've applied a high-content single-cell RNA sequencing approach to our bioprocess. If you take the bioprocess and you know, with the expertise of Lawrence and his group, what is a good midbrain dopaminergic neuron, and you run that process many times, and you isolate samples along that continuum at various chronologic time points, and you analyze the single-cell RNA-seq pattern from individual cells, tens of thousands of cells or hundreds of thousands of cells in each of those preparations, and then you compare the profile you get with the output from the process, you can start to understand what might be correlated with a good process. We've done that. You can run these AI approaches to cluster the data, and what you find is, in this case, 5 separate clusters. You'll just have to, the colors and the location tell you a lot about the cell and its expression profile. And from that, you can derive from this highly complex and rather complicated approach with AI down to a definitive set of genes that are a panel that predicts where in the process your cell is, as far as whether it's progenitor stage, it's early neuron stage, it's definitive midbrain dopaminergic, or whether it's an impurity. And from that, you can tune the process. So we've taken this, what is effectively billions of data points and a highly research mode, and we now have high confidence that you can use that approach to come up with a simple digital test to follow your bioprocess. So it's, I think, the first example for me where AI really is going to make a difference, and it's a broadly available tool in single-cell seq. So the question is, can you do this? The 2 challenges in the industry are, how do you test for potency in cell therapy, and how do you test for tumorigenicity or safety risk? Both of those are 9-month bioassays that take effectively a year and a half to run. And the question is, how do we all avoid something like that? Because it's expensive and slow. So if we took this same approach of an AI approach in single-cell seq to look at the correlation between the population of cells that we produce and their outcome in vivo. We run a process, you take the cells, you analyze them, and then you compare that to its engraftment and response in the animal. And if you take a population of 15 of those runs where you know what happened when you put it in the animal, and you know what the single-cell seq profile looks like, and you build a map from that, you start to see these signatures, and you can start to identify cells and correlations of signatures that map to engraftability and to potency in vivo. From that, what we can do is, this is an example where we took the next run, we took another run, and ran it against the AI and said, did it correctly predict the potency of that batch? And what we see is that it did. It put that little blue dot on the green side of the plot, meaning that the AI predicted that this was a potent batch of cells. So I just show you this because it is the first example for, I think I've seen, where you're starting to see an in vivo bioassay as complex as potency get replaced by, instead of a year and a half, a week and a half test that's digital. So we think it has tremendous promise. Similarly, the tumorigenicity test, which is the bane of probably everyone's existence in this room, it's a delay and it's expensive. How can you test for tumorigenicity? Is there an approach that could have merit? So we asked whether you could use AI to detect rare cells. So the fundamental premise is that if you have a tumorigenic risk, it's because there's a cell that will proliferate rapidly in vivo, and that it's probably a very rare cell, on the order of 1 in 10 million cells, let's say. And the problem with that is, a mouse, you can only put so many cells into a mouse, 100,000. So if you have 100,000, you need 100 mice just to test for 1 cell. It's a practical impossibility. So you need a better way to do it. The FDA knows this. There's a whole team, lots of people working on this problem. We think AI and the ability to detect rare cells, there is a way to do this, maybe. As this is one of the approaches I know many people in this room are probably taking as well. So what we did was to take our population of terminal cells, to spike in another cell, a different cell, at limiting dilutions, and then to take and stain for that cell, and then to ask a technician, highly laborious, to go through and scan for positives, false positives and false negatives, scanned totally subjectively by the technician. Then we put that same data set without the subjective coding into the neural network and said, can you predict the presence of a positive and a negative? and build from that an algorithm that says whether the AI can predict the presence of a cell. And in this case, you can see the technician scoring a false positive or a true positive. You do that with a very large data set. And what you end up with is, and then you take that output of the AI and you ask whether it can accurately predict the limiting dilution set. How many rare cells does the AI predict are present in your population? And what you see is the dark blue and the light blue bars showing that in this experiment, in this engineered test, the AI perfectly predicted the number of cells present, the rare cells, which in theory could be used as a prediction of, for example, a pluripotent cell in a tube of 10 million neurons. What's exciting about that is it would allow you to test the full human dose. The 10 million cells on a dish is not that big a deal when you're screening a two-dimensional substrate. So I would announce we've recently completed our pilot GMP facility in Toronto. We're very excited about that. We're in commissioning and validation, and we will be using this for all of our early stage manufacture. It's a unidirectional flow facility, gives us the ability to control our own destiny in that early stage manufacture. We're also building a commercial facility with the help of our new Bayer colleagues, and you'll be hearing about that coming soon. But that will help us to meet an accelerated RMAT designation timeline, we hope, and to also manufacture our materials in-house for the lifetime of the product. So last thing I'd say is, if I skip this, is just to say that if you have availability in November, please come to a meeting we're co-sponsoring with ISSCR. It's a translational cell therapy meeting. I think you'll find a spectacular list of presenters, and we're very excited by it, November 6th to the 8th in Toronto. Please come join us. Other than that, thank you for listening, and I'll be around for questions.